So I hope everybody can hear and see me. Good morning, good afternoon, good evening, depending on, and good night, depending on what part of the world you are in. A warm welcome to all of you. So let me just introduce the seminar series and its objectives, although most of you are familiar with it. So critical transitions in complex systems seminar series is being jointly hosted by IIT Madras, Indian Institute of Technology, Madras in India, and the Potsdam Institute for Climate Impact Research in Potsdam, Germany. So this seminar series brings together experts from a variety of fields because complex system is an interdisciplinary topic. So a variety of fields such as climate science, neuroscience, fluid mechanics, combustion, ecology, etc., and aims to disseminate the state of the art in the prediction of critical transitions in diverse fields of study. So the seminar takes place every month on the last Monday of the month. And we typically have a seminar for 9 months, and in summer we do not have. So this would be the last for this season, and then we'll restart in September. Before we introduce the speaker, a few housekeeping notes. I request everyone to turn off their microphones so that the seminar can proceed smoothly. Please use the Q&A box. There's a Q&A box if you look at the bar in Zoom, and you can type your question there, and the speaker will answer all your questions at the end. We have plenty of time at the end to discuss all your questions, doubts, and clarifications. Now I request Professor Jürgen Kurz to introduce the speaker. I think he will do it without the slides. So, okay.
Yes, only. Yeah, thank you very much. Can you hear me? Thank you very much, Suchit, for the introduction and for the main organization you and your team are doing. And today we have really, again, a very interesting and very important speaker in our series. This is Professor Gabriel Mindlin, coming from University of Buenos Aires in Argentina. It's a place very far away from India, from a topographical viewpoint, also not so close to Germany, by the way. And he is an expert in different aspects of nonlinear dynamics, especially in bifurcation and such things. But what he then has done in the last, I would say, decades already, is studying the birds, especially the singing of the birds. How are they doing this? And now even going to the dreams of the birds. And this looks a bit poetic, but it is based on really very hard neuroscientific approaches, very hard measures and very interesting models where he can explain a lot of phenomena of birds. And I'm really very happy that he accepted our invitation, and I'm looking forward to your talk, Gabo. Please, you have the floor.
Thank you very much, Jürgen. Thank you all for the organization of this webinar. For me, it's very important to be able to tell these stories coming from so far apart. So it's a real treat for me to meet you all. Let me see if I can start my presentation. And I'm going to be acknowledging all the co-workers that participate in this research at the end, because, as you said, this covers many years of work. So let me start doing some review and jump to the latest results on listening to dreams, which is the topic of the title of the talk. No? So in general, I could say I'm going to be talking about the physics of birdsong and how to use it to listen to a bird's dream. And this is work I've been doing mostly at the University of Buenos Aires with a variety of co-workers. So why being interested in dreams? That's a very easy thing to answer, because dreams are some of our most intimate and elusive experiences. So we all fantasize in a way with the possibility of recording a dream, having a dream which we cannot recall, but somehow recording it and reviving it and reading it again and watching it again the next day. And of course, we're very far away from doing that. We cannot do this right now. And the main reason for which we cannot do that is because we don't know the code used by the neurons to codify behavior, perceptions, and stuff. So there are many advances. There are thousands of neuroscientists working in every aspect of this problem, but we still don't know the code to the degree to which we can interpret a neural pattern and translate it into a sound or a figure. There are some advances using artificial intelligence that I can discuss at the end, but we're still far away from solving this problem. So before continuing, you are listening to me. There's no problems with the sound.
No, we can hear you.
It is perfect.
Perfect. Okay. So in order to advance this program, I'm going to borrow some ideas of neuroethology. Neuroethology is a wonderful field of research. It studies the neural basis of behavior. And they have this concept, which I like very much, which is the idea of a champion species. Because I'm usually asked, why are you studying birds, or why are you studying this particular species? And this concept comes from neuroethology that says that if you want to study some behavior, you can be sure that there is going to be some species that has spectacularly displayed that behavior, or is particularly easy to measure in order to unveil the mechanistics behind that behavior. So for example, ocine birds, birds that are called songbirds, they constitute more or less 40% of the known bird species, and those are the birds that have to learn their vocalizations, like zebra finches or canaries or cardinals. They have been used like an animal model to understand how a complex behavior like vocalization is learned. So how a brain configures itself in the process of learning a complex vocalization. You can study that wonderfully in birds, in songbirds, particularly because you have brain areas that are specifically dedicated to that. Okay, if you want to study the problem of vocalization in humans, it's a mess because all the regions are completely integrated in a very complex way. In birds, you have nuclei that are specialized in the generation of the song, in the correction of errors, and so on. So why this is important if you're interested in dreams? The reason is that due to the need of unveiling the role of sleep during learning, there's been a substantial work in studying the neural activity in different parts of the avian brain, okay, when the bird is sleeping. So a seminal work was performed in the year 2000 by Dan Margolius at the University of Chicago. I would say that the first leading guy in that. And this guy, so what he did is the following. He measured the neural activity of a bird when the bird was singing. You will have some specific pattern if you are measuring some particular area. And then he noticed that at night, when the bird is sleeping, if you measure in the same neuron, this is a high impedance electrode that is measuring just one neuron, eventually you can see during sleep the same pattern of activity. So the bird is completely asleep, is silent, of course, but then at night, eventually in that region of the brain that during the day was active in the production of the song, has this replay, this silent replay. But of course, the issue is that you don't find only patterns that are very similar to the ones that are used during the day. In those same regions, you can have also non-veridical ones, things that the birds would never use during the singing. And here comes the problem. Now, you don't know how to interpret those, no? Because we still don't know the code, even for birds, of how the bird is coding its complex behavior. But at this point, I would say that physics and dynamics do have something to say about this. So what I'm going to be doing is describing very briefly the physics of birdsong, how this can be used to study the problem of dreaming and its connection to learning. So in a few seconds, how birds generate the songs. Okay, this is very simple. This is a movie, an animation done by Roderick Suthers a few years ago. They have an avian vocal organ, which is the syrinx, is a bipartite device located at the juncture between the bronchi and the trachea. And we see here during normal respiration that the air is coming in and out. But then the bird does something, changes the configuration of the vocal organ, the labia come closer, and they will start to oscillate. Very similar to the vocal folds when we humans produced a voiced sound. And while they are doing it, they are changing also the tension of those vocal folds or labia in order to change its elastic properties, restitution constant, and therefore the frequency of those oscillations. So what the bird is doing basically is generating some modulations for the airflow that are going to be passively filtered in here, okay? And the more nonlinear aspect of this problem comes from studying the dynamics of these labia, which are set in motion when you pass some air in between them. So basically, if you write a very simple model for the dynamics of this labia, which will be writing basically Newton's law for the midpoint position of a labium, you will have that this would be the midpoint position of the labium, and you will have the forces, and you will have a restitution because it's an elastic material. You will have some dissipation. You have some nonlinear dissipation because when the labia collide with each other or with the walls, they're going to dissipate a lot of energy. And you can compute which is the average pressure when you have some particular combination of modes in terms of the labial motion. So both the air sac pressure, okay, which controls the airflow, and the restitution constant of the labia that depends on how stretched these labia are time-dependent parameters that enter into the equation. So again, these seedings has this huge labia, the syringalis ventralis. When they are contracted, the labia that are inside get stretched and the frequency is modified. So by modifying the air sac pressure and the tension, you can have a range of possibilities for the sound that is being generated. So as Jürgen was saying, during the years, and together with Franz Goller, who was then at the University of Utah, we've been measuring these physiological instructions when the bird was singing. So you could insert a small cannula in one of the air sacs and transduce that into voltages, and also through some delicate surgeries, you can implant some tiny electrodes in the muscles, and when the bird recovers, you present the bird with the female and the bird will start to sing, okay? So you can record all these physiological instructions at the same time that we're recording the sound. Very briefly, and summarizing this, you have this nonlinear device which modulates the flow, which is ultimately filtered by a passive Helmholtz oscillator, basically a trachea. This complicated, or more or less complicated, low-dimensional model can be translated into a normal form, and you can eliminate many terms, many unnecessary terms, and really find the skeleton of the dynamics. And this bifurcation diagram that summarizes the dynamics of this normal form has all the dynamical elements that are necessary to build the acoustic properties of many birdsong species, songbird species. As I was saying, the pressure and the tension, which are time-dependent parameters, are going to be determining how you go around this parameter space in order to generate these oscillatory motions and stuff. But again, if you turn the oscillations on close to a Hopf bifurcation, you have a very simple harmonic signal. But if you turn them all in a saddle node in limit cycle, you will have some very spectrally rich sounds because you have this explosive motion. So you have a variety of sounds that can be reproduced even with this very simple model. So simple that if you compare the sound that you generate with by the bird with the one that you can synthesize with the normal form, I'm going to be playing 2 songs to see how similar or different they are. Pay attention, this is a very complex song and very short, so you have to be very, very attentive, okay, because it will last a second or so. Okay, this is the song. Okay, it goes again. And this is the normal form generated the sound. Okay, so you can with a high degree of dimensionality reduction generate things that are similar. We'll discuss later how similar they are, how pertinent they are. But some interesting thing happened collaborating with Franz Goller. He was measuring several muscles simultaneously, and here you have like the air sac pressure and two different muscles when the bird was singing. Okay, the different colors are denoting different syllables, so you have the specific patterns that are used for generating a syllable, and you see they're repeating if the syllable is the same, etc. But one time he had one bird that was connected. He inspected what happened at night, and the discovery was something very interesting. We saw that in the first slide, that in the brain, eventually at night, you will have some replace, so patterns of activity in the brain that were similar to what the bird was using for generating the sound. And what we notice is that at night, the bird was having the normal quiet respiration of a sleeping bird. It was completely silent, but from time to time you would have these patterns of activity that were very similar to the patterns of activity that the bird was using during the day to generate the song. So this means that whatever is happening in the brain doesn't go through the respiratory system, gets inhibited somehow, but the part that is going to be affecting the vocal apparatus doesn't get stopped, and you can measure these practices in the masses down there. And the good thing is that once that you are in the masses of the vocal organ, you have all these mathematical models that allow you to synthesize sound. So you don't have the code to interpret sparse activity that you're measuring in a part of the brain, but once that it goes down to the masses, you can translate that into behavior. So the advantage of using it so closely, measuring so closely to the periphery, is that now you have a mathematical model that allows you to interpret what those practices are all about. So just to see what kind of things happen through the singing part of a brain of a bird when the bird is sleeping. Here you have the pressure, okay, when the bird is singing during the day, the muscle activity patterns that are associated to those pressure gestures. And if you chunk, okay, pieces of this muscle activity, you can take the activity that you measure at night, where you have from time to time all these crazy activity patterns, and see whether some of these correspond to actual patterns that you see during the day. And what you find, and this is very interesting because these birds, for example, zebra finches, the one I'm studying here, they have a very stereotyped song. Once that they learn only one song, and they repeat it over and over. But at night, they do more, okay? They have the practices of what they use, but they also have some junk, if you want, some non-veridical practices. So here's a summary of what happened to this one bird. Like more or less each hour is going to be practicing something, and with different colors, I'm coding which gesture, okay, the bird is practicing. So for example, in the first hour, it practices them all, but then during this hour, notice that it's practicing mostly this gesture in here. During this hour, it's practicing this gesture in here, okay, and so on. If you do the statistics of this, most of the time it's just practicing either one syllable or a combination of two syllables, and just at the end of the night, maybe some time, it will practice the whole song. It's more or less like when you practice an instrument and you practice a difficult passage and then another difficult passage, and then at the end of your musical practice, you practice it all together. But what I'm going to be focusing today is in a question that we had, that was how much of this is really associated to learning or not. And this is the beautiful thing about studying birds. You have 10,000 species. So if you have a question like this, you can use another champion species to address this question. So what I'm showing in this graphic here is a phylogenetic tree of all the bird species. And these red ones are the songbirds: canaries, silverfinches, cardinals, thrashers, and so on. These are vocal learners, okay? This is a suborder of the passeriforms. The passeriforms include these, and they also include sabocines. And sabocines are birds which I love, first of all, because they are very represented in South America, so I can study them easily and cheaply. But also they are phylogenetically very close to oscine birds, but are suspected not to learn. So they don't need to learn in order to have their vocalizations. Okay, they are imprinted in the genes. So if you want to study a question that is related to learning, it's wonderful to practice in comparison what happens with the learning bird and a non-learning bird. So the main character I'm going to be describing today is the kiskadee, which is a non-learner. And this is the kind of vocalization that it does. It's a very simple sequence of calls, okay, kiskadee, benteveo. In every country is called in a way that onomatopoeically reminds you of the song of the bird. And if you measure the activity of the masses, here comes the first surprise. Well, it's not a surprise to us now, but it's kind of surprising for many different species. The strategy of how to use your muscles is different. What I mean is that in oscine birds, what I described before is a very well preserved structure. But in non-learners, when the birds cannot learn, or they cannot do these sophisticated modifications of their gestures, the way in which they find their niche in the acoustic landscape is by anatomical modifications or by uses of the vocal organ that are different. So for example, in kiskadee, it took us several years to unveil what it was doing. What the birds do is this, they modulate the frequency with the air sac pressure, but they use these huge muscles that are very similar to the syringalis ventralis muscles in order to keep the two sound sources out of tune. It's very interesting. So if you measure the EMG activity, what you find is that it has a low frequency modulation, like maybe 100 hertz. And it corresponds to something that we observe in the sound, that is a 100 hertz modulation. You have a very high correlation between what you see in the EMG and the amplitude modulation in the sound. But basically what it is the following. They have two sound sources that are slightly out of tune, okay? Out of tune in the order of the 100 hertz. But when you force them, if you force them at more or less a frequency within that order of magnitude, like 100 hertz, 120 hertz, you lock the two sound sources to keep exactly out of tune at the forcing frequency. So you use the forcing frequency to make sure that the two sound sources stay out of tune in a precise amount. And being out of tune gives this roughness, which is a little bit intimidating, actually. Okay, you avoid being very tonal. You sound kind of very rough. So, for example, this is an awake bird. This is the EMG of the awake bird. This is the sound generating by the awake bird, and this is the spectrogram. So it's the musical script of the sound. So you have the frequency in the vertical axis and the time in the horizontal axis. And when the bird is asleep, you measure eventually something that is similar, maybe a little bit slower, okay? Nothing in the sound, of course. So what we decided to do is to use this EMG activity, plug it into a model, okay, and generating the sound that would correspond to that EMG activity. So if you take the model of the sound sources and you plug in terms of the tension, okay, this EMG, what you do is you generate a synthetic sound. So let me see how it sounds, a typical call when the bird is singing, okay? Okay. And this is what it sounds when you use the sleeping EMG in order to generate the sound. Oops. Okay. So it's very difficult to see the difference when you are not used to it, but the modulation of the frequency is a little bit lower. Also, the timing of the syllables is different in the Kiskali. But this is not the only vocalizations that the birds do. When the birds are not doing the call, they have a second kind of vocalization, which is a very intimidating trill that they use when they're upset. For example, someone comes into a territory. So what I'm going to be showing you is a movie of a bird that is finding another bird of the species in a cage and goes to make sure that it's not invading. And you will see that it makes some trills. And together with the very interesting display of feathers coming out, and gives a very intimidating aspect, okay? So there is a tiny dolphin-like trill together with this display. Interesting enough, when you record these guys with an infrared camera, the bird is sleeping, and from time to time, you see the display of feathers coming up. So we measured the activity when the bird was doing this display, and it was very interesting to notice that when the bird is doing this display, the masses would have a trill-like activity. So it's a different kind of vocalization. It's not a long 200 or 300 milliseconds vocalizations, but it's more like a repetition of 30 or 40 millisecond syllables. So, of course, we couldn't resist the temptation to synthesizing them using the images in the trill, and you have a trill that sounds very similar to the trill of the bird. So, what are we planning to do with this? Can we manipulate dreams? And what would happen in terms of learning if you manipulate the dreams? Let me just, before continuing, that finding dreams in Osine birds was very interesting because we were planning not to find them. We were very convinced that the replay at night was associated with learning. So to us, it was a surprise to find that non-learning birds do practice at night. So it's a very natural question, why are they dreaming? The answer is we don't know. But one interesting aspect is that when an Osine bird, a songbird, the ones that learn, practice at night, they have what they use, and they also explore around a lot. When a non-learning bird has the replay, it just has the replay very similar to the few vocalizations that they have. So we don't know which is the purpose of the replay, but we do know that learners have more complex replays than non-learners. So the idea to me is that it would be very interesting to use the possibility of generating sounds associated to the dreams in order to explore different questions. Like, for example, what would happen if instead of non-veridical dreams, you would urge a bird, force a bird to have a stereotype dream? Would that affect the learning? Or what happens if the bird that is having these crazy non-veridical practices would actually listen to the sounds that are produced with it? So how that would affect the learning. So in order to proceed with that research program, a first element for us would be whether we can induce some neural patterns and muscle activity patterns at night. Okay, can we induce a dream? Okay, so let's see. When the bird is singing, okay, here we have a bird that is singing. This represents the sound that is being sung, and if you measure the the muscle activity, you have this complex pattern. Okay. Now the bird is sleeping, and we are playing, okay, back the birdsong song. And what we find is that in the masses, you recreate a pattern that is very similar and highly correlated to the pattern that was used during the day. This is somehow not surprising because Dan Margoliouth, working with Konishi, much later, and Amador, many times it was used this idea that if you play to a sleeping bird its own song, you would elicit a response in the part of the brain that is associated to producing the song that is very similar to the pattern that was used when the song was produced. It's highly selective neurons in the song system are sensitive to the auditory stimulus of the birdsong song. Now what we measure is that this replay that you can induce, okay, goes down to the settings. But not even that. If instead of playing the bird its own song, you play a synthetic sound generated by our low-dimensional models, you also can induce activity patterns very similar to the activity patterns that are measured when the birds are exposed to the birdsong song, which are, by the way, similar to the patterns used when the bird is singing. And it's not a trivial achievement because if you play something else, like for example, a reverse song or a specific song, the song of another zebra finch, you have no response at all. But if you have a synthetic copy, you have a very good copy. So the idea is, can you actually induce a song? Can you override can you induce a dream? Can you override what the bird is doing? If it's doing nothing, you can. Okay, you play the bird's own song or a synthesis, and you are going to have an activity pattern similar to the one used in song production. But now what Facundo Feinstein, a graduate student in our lab, did is the following. He identified segments where a canary, which is sleeping, would be starting to have a replay. So typically they have long replays of several syllables, but when he identified that there was this starting of a spontaneous practice, he played the song. Actually, he looked for this happening by chance, but anyways, the experiment could have been designed like this. You measure this spontaneous syringeal pattern, and then you start to play the song. So what you see is that instead of continuing with the replay that the bird already started, the bird locks and starts to display the syringeal activity that would correspond to generating that sound. In other words, you can use the fact that the bird is starting to dream to override the dream by playing the bird's own song. So to us, this opens a very interesting gate to the problem of interpreting what is the role of the dreams in terms of behavior. Because what we are designing to do is, first of all, to extend this model of synthesizing dreams to assign birds. Just a minute. Okay, thank you. To extend the synthesis of replace to assign birds, subassigns are simpler. Assigned birds are a little bit more complicated because you have not only to take into account the frequency modulation, but also the gating. We're very close to nailing that. And they were planning to expose a sleeping bird to the synthesis of its own dream. And we plan to do it with learning juveniles in order to see how that affects the learning. We also plan to do the following, to trigger a stimulus, that is the birdsong song, when a sleeping bird starts to dream. So we want to do the following. Instead of letting the bird dream, a very non-veridical practice, each time that the bird starts to dream, we're planning to induce a stereotyped song and see how that affects the learning. So we want to generate this, we want to use these synthetic models of birdsong production and synthesizing dreams in order to explore why birds that learn need to learn crazy stuff and why they do the practices that are crazy silently, okay? So these are the two questions that we want to address. So let me tell you the conclusions, and then we will open the time for questions and comments. So my first conclusion is that usually in neuroscience, you study neural patterns, and you study it more or less isolated from actual behavior in a way. I love studying all that together and using physics and dynamics as the bridge between these neural patterns and behavior, because behavior emerges from the interaction of a nervous system and an environment, and physics plays an important role in that. So I love the idea that physics is used like to poke what's happening in the brain. And the reason for which that happens is that at the moment in which a physical device is commanded, you have to have the general readout of the brain, okay? When you go and look for high impedance electrodes, sometimes you see in detail a small picture, but then when you want to link it to behavior, it's good to have a global readout, and this is what you have at the periphery. So this is an example of the kind of nice things that you can do if you look at what happens in the masses and understanding how the masses are translating these activity patterns into behavior. Also, sometimes it's not just making a very complicated and detailed physical model. Sometimes a dynamical model that captures the essence of it is enough to understand very subtle characteristics of behavior. For example, if your oscillations in the labia are ruled by a subtle node in limit cycle, then you're going to have very spectrally rich sounds, and you can generate very realistic sounds without needing to have a very complicated high-dimensional dynamical model. You can have a low-dimensional model that, capturing the right bifurcations, and that is enough to cheat highly selective neurons in the bird's brain. To me, that's also a very interesting conclusion. So the other one is that we have an operational way to read dreams. I think that this is not a small thing. We tried, of course, like we do always, with the first and simpler species, but then if you go to assigned birds, you will have a very complicated vocal apparatus, and you will have a way to translating whatever comes to the brain into sound. And well, I guess that's an interesting tool, as I was describing at the end of the talk. And then we're collecting questions. Why do non-learning birds do practice? Why do learning birds practice outside the stereotypy? I think they're very interesting questions in the purpose of dreaming and how it relates to learning. And so I was saying before, what happens if we don't let crazy dreams or non-veridical dreams be part of our learning experience? Do we achieve the same excellence in the performance of our actions if we don't practice crazy things at night? So that's something that I guess it's interesting to pursue. And also the following, maybe you need this non-veridical replace to explore beyond what you have already achieved. But what happens if you do have feedback for those? Does it affect? So why you need to do crazy things silently? I think this is another very interesting question. And of course, a program so long wouldn't have been possible with the help of many people. Some of them are not listed because they participated in much earlier stages of all this program. But right now, I would like to thank Anna, of course, who runs the lab with me and is responsible for all that is real neuroscience in our lab. Franz Goller, who has been very generously helping us during the years, and he's an expert in physiology, in muscle physiology in birds. Dan Margolias from the University of Chicago, who is a wonderful collaborator, one of the leading experts in the neuroscience of birdsong production. Some of the younger researchers that were more involved in this research are Juan Doppler, who did his PhD on this Kiskadi work, Facundo Feinstein, who is listening to canary dreams right now, Alan Bush, who participated in the induction of dreams at the beginning, Melina Tencio, who is a biologist, a real bird specialist, and other members of the lab, Roberto, Leandro Fiamma-Hernan, and Agustin, who are helping us pursue all these lines of research. And I would like to thank you for your attention, and I would love to discuss some questions.
Yeah, thank you very much. Really very exciting talk, Gabo. And I would like to hand over to Gaurav to manage the QA part.
Thank you, Professor. Professor Gabriel, that was a very interesting talk. You opened up a new area for us. So beneath in the lower window, you can see the Q&As. So do you want me to read them or can you access them?
Okay, no, I could access them, right.
Okay. Okay.
Okay, so the first question is, do some birds keep learning all of their lifespan or only during adolescence? Okay, that's a very nice question. So the first thing I would like to remind you, Devjit, is that there are 10,000 species. So there is a variety of strategies in terms of learning. In general, they all share this scheme, this pattern, where when you are very young, you are exposed to the sound of a tutor, then you are babbling for a while, and then you practice. And once that you practice, you crystallize the song. Yet there are species where every year there is room for reopening a little bit the window of learning. So for example, zebra finches, they would only learn at a specific time of their life. It's, in your words, during adolescence, if you want. So they will have a very brief period where they are open for that. And once that they crystallize, they practice, they sing only this one song. One interesting aspect, to me at least, is that in that case, for example, during its whole life, it's going to practice, it's only to sing this one song, but at night it will continue having variability. So variability is not only necessary for getting the song, but maybe also for maintaining the song. But then if you go to canaries, every summer, you will see that probably if you had a pet, a canary as a pet, or any family of yours had a canary as a pet, you might have noticed that during the summer they stop singing, okay? And then when they start again, they might have incorporated new syllables. In that case, there's something very interesting, that is that the hardware gets updated too. So you have neurogenesis, you have new neurons that are generated and they go to the song system and they become functional. So in that case, they will open several times during their lives the window for learning. Okay, and then there's another one. Are there signals we observe when a bird dreams simulated by some other part of its brain, stimulated by some other parts of its brain? Also, do the perceived parts of the brain show activity in response to its own dreams? Okay, so the second one, okay, let me see. selectivity, okay, let's see. If I interpret the question correctly, let's say, suppose that the bird is listening to its own song. In that case, what you would have is activity in HVC, for example, that is a region that integrates auditory pathways and the motor pathways. Now, when the bird is only dreaming, I don't know of anyone who have measured something different from the song system. So it's HVC, let's say RA, so all parts of the song system. Auditory parts of the system where the bird is having spontaneous replay, I don't think anyone has measured, and it would be interesting to do so. I don't think anyone has. So it's related to the first part of the question. So when you have spontaneous replay, I think we've only measured things in the song system, not in perceptive like areas, like area L or other auditory areas. So that hasn't been measured. The third one is, how about the effects of environment on their songs being produced? Can the songs be sad or happy? Well, this is a very interesting question because I would say that many, let's call it emotional aspects, can be translated into some song features. For example, if you take a zebra finch, it will practice this one song all the time, all day long. They do nothing else but practicing this one song. And this one song has one purpose, to impress a female. So when you present the female to the song, it will look straight at her eyes and start to sing the song. But the song is slightly different. It is more precise. It will repeat it over and over with the precision of a machine gun, okay? So when it's just practicing, it will have some variability. When it's addressing the female, the timing gets more precise. So there are features of the song which are, have a fingerprint of their emotional state, if you want, at least in terms of arousal. And there is a lot that we don't know about that. We are just starting to scratch the surface of animal communication. So if it doesn't relate to something we're expecting, it's difficult to find. So people are concentrating on syllable combinations, acoustic features of some syllables, but these more holistic features that can be reflection of emotions, we still haven't done it, at least outside this example that I can quote, okay, the timing associated to some interest that is sexual in nature, in this case. Are these dreams like humans? Are the lucid dream stage where this can be manipulated? Well, that I don't know. That we don't know, actually. There is a lot that we will have to be doing in this program. As you mentioned, one of the first things would be to separate the night into the different stages of the dream. So Anna, with a student called Manuel, are designing the electrodes to measure in which part of the sleep stage the birds are, and how that relates to the different practices that they do, the different replays that they do. Because ocine birds share with humans these stages, not like REM phase and so on. So this correlation is still to be done. So I guess that is an open question. Oh, there are more. What do you expect to see when you expose a bird to its own dream synthesis? Well, I'm dying to, no, I don't know. To tell you the truth, I don't know. And I'm very curious. So let me tell you what the experiment is about. When the birds are learning, when the juvenile is learning, the quality of the song at the very early in the morning changes along the day. So the song improves, okay, along the day. Then you have the night and you start all over again. It starts a little bit worse and then it gets better and then so. But the amplitude of this effect gets smaller when the bird is crystallizing the song. So what I would expect, or what I would like to test, is whether when the birds are listening to their own dreams, this modulation changes. So I'm planning to see the effect on listening to their own dreams in terms of the modulation of this effect during the learning. This is the actual experiment. Because there is this conjecture that this effect of worsening the quality that you achieved the day before has to do with the fact that you were having practices that didn't have auditory feedback. So we will test that hypothesis. So my conjecture would be, or what I would expect, is a change in this modulation of quality that you see the next day after a night of sleep. If you are not exposed to a sound, and it's a silent practice, it is what we have measured. If you make the bird listen to its own dream, I will expect some change in the modulations of quality in a day-to-day basis during the learning.
Professor, I would like to ask Professor Kurz, would you like to ask any questions?
Yes, I have a
Yes,
question. The first one is
please.
already answered, this REM, non-REM. But the other question is, you mentioned already that the birds are singing in order to attract females. Yeah? And I think this is very typical in nature. And what are then the criteria for the female to find it especially attractive?
Beautiful question. So, to me, this is the key question. So, what I can describe is what we're doing now in the field with this. So the question, Jürgen, is what is sexy in a song, right?
That's in other words, yeah. Yes.
Right. What is sexy in a song? So what we're playing to, so what we've been doing is the following. We've been studying bird species, which is called the chingola. It's a Sonotrichia capensis, which is a songbird that is very frequent here where I live. So what we've been exploring so far is how they react to synthetic songs, and basically what they do is when you put a device that is broadcasting a synthetic sound, they go crazy. They look for the invader and they try to take them out. So what we are designing now is a set of experiments where you change some features of the song to try to explore what they find more intimidating and also what they find more attractive, the females. And we don't know. But the hypothesis is that what they find are males that are extremely skillful at doing some difficult things. So, for example, if you have a bird that has repeated syllables, there is a law, basically, because of the limitations of the physiology, that if you want to span a wide range in frequency, then you have to have a very a long time between syllables. As soon as you start to increase the rate of syllable production, then the range of the frequency that you can span for each syllable has to be smaller because the speed at which you can move the masses has a boundary. Okay, so the product of rate and frequency modulation is bounded. So the hypothesis, the most credible hypothesis that has been put forward is that females will find attractive the guys that can have wider modulations at the most rapid rate of production. This would display a very skillful motor device. So what I want to do is to generate these synthetic copies with this model and go to the limit and see whether more females get attracted. And then I would like to go beyond and see whether freak is sexy or not. So I don't know. we are designing these experiments with a student called Roberto Vistel right now. But I would say that the skill of generating wide range of frequencies very rapidly would be a key for sexiness. It is my conjecture.
Thank you very much. Very great. Okay.
Yeah.
Uh, um, Professor Ambika, do you have a question?
Yeah, I was just wondering, there are birds like parrots that can produce and reproduce a large number of signals. They can even reproduce human speech. You think it is because of a difference in the muscle structure? Is it more complicated than these birds singing birds?
So yes, that's true. So the birds that can generate very complicated copies, like the ones that can copy a car or an alarm car and so on, I still didn't study any of those cases. So all I could do is to share conjectures of no value, so calandrias and so on are wonderful examples, and I don't know actually what happens with those species. What I could tell you is that cases like sparrows that have this ability to generate human-like sounds, is that they have an upper vocal tract that is very different than songbirds. So now you're talking about a different order completely. In this organization that I was showing, you are not in songbirds, you are not in sabocines, you are in a completely different order. And there you have different features of the vocal apparatus, like having a tongue, for example, that allows them to have these rrr kind sounds and stuff like that. And to tell you the truth, I know that there are these differences. It's a very exciting problem, but I haven't addressed it. So to summarize, there are two different aspects. One is how some species can have very complex repertoires, while others, like zebra finches or maybe canaries, have more bounded repertoires. And this is a question for neuroscience. How is the neural architecture, the neural network that is able to produce a wider repertoire richer than the other ones? This is one question. The answer is, I don't know. The other question is, how is the vocal organ of the species that can reproduce the sounds? In the case of parrots, it has to do with tone and differences in the upper tract. So, all beautiful questions, no answer for any of them.
Thank you very much. Yeah.
Professor Gabriel, I have a short question.
Yes.
Is this dreaming, like connection between dreaming and the vocals, is it something vestigial in birds? Like, was it useful in a previous, like prior to evolution, or now it has become, yes, less useful? Is it vestigial kind of thing?
So I don't know. The question is complicated because you see, when it comes to vocal learning, we humans share vocal learning with just a few species, okay? So some mammals, like cetaceans or maybe elephants, some bats and so on, and then there is a huge desert until you go to birds where you have this vocal learning. So they could be independent solutions that appear and are not connected directly. So to do the mapping, the evolutionary mapping, it's kind of complex because sometimes you have preserved features, but in other cases you have independent appearance and disappearances of some abilities. So this seems to be the case for vocal learning and maybe also for dreams. I don't know.
Okay, yeah, thank you. There is one question in a webinar chat. I'll read it out. Are there instances of extreme events or large variability in the song of a bird, especially during the day when it is expected to only repeat the song? For instance, some accident like destruction of its nest can excite an extreme reaction from the bird.
Is it written? Can I read it? It was a long one.
Yes, it is in the chat. If you can see the chat.
So humans REM captive and gender.
No. In the chat, I have
Oh, in the chat. Okay, in the chat. Sorry. Let's see.
by S. Shruti Tandon. I'll just try to copy paste it.
Yeah. Well, this focuses to something very interesting, that is the power and benefits of doing field work compared to lab work. Because what you're asking, that is very interesting, is whether some life sensitive event, like an extreme event, will generate some variability either in the behavior and stuff like that. So the answer is we don't know. And the kind of experiments that would be needed will be very rich experiments where you will be able to do these experiments in the wild, probably when the bird is exposed to behaviorally more challenges than what you do when you work in the lab with songbirds, okay, where their lives are monotonically very controlled and so on. So it's a very interesting question, which I have no answers for. Yeah.
Okay, thank you. So I think there are some more questions in Q&A.
Yeah, there was one about gender. That's very interesting.
Okay.
And captive birds and birds in captivity. Both very interesting questions. So, gender. In most bird species, is the male who sings and the female who decides this key question that Jürgen was asking. That is, what is actually interesting to check in a maize display in order to select who is going to be the father of the next generation. But there are some species where both sing. And also there is a very interesting species that we are studying, and Anna actually studying, which are sabocins. In Argentina is the hornero, is Furnarius rufus, and it's a species where both male and female sing, and they engage in a duet which has a rhythmic complexity which is astonishing. You see devil staircases and locking and synchronization, and you have all sorts of very rich dynamics in between the vocalizations of the male and the female. And interesting enough, Anna and her student Thomas are finding that there is some degree of learning, some elements of learning where you have some richness that hasn't been detected before. And the richness comes precisely in the vocalizations of the female mostly. So whether there is a statistical problem of a gender bias, it is true that female vocalizations have been studying less, but they will be giving surprises probably in the next years. Concerning birds in captivity and birds in the environment, okay, so basically you have studies that have been done on specific species that are bred for laboratory research, and you have wild birds. We have worked with wild birds for this Kiskadi experiment, and working with wild birds is very complicated because mainly it's very difficult to do it, it's very difficult to get the permits. When you do experiments with wild animals, you can keep them in the lab just for a short time, then you have to have a whole protocol of returning them and so on, and most of them don't adjust to laboratory life. So not all the behaviors are displayed in the lab. It's exactly as you say. For example, we could elicit the normal songs, but the birds that we had in the lab, we could elicit at night these nightmares, if you want, these trills where the birds were having the feathers coming up and this muscle activity of the trills, but we could never elicit a trill in the lab. Probably they were intimidated, they weren't having a good time, so we had to release them to have them display the normal trill naturally. So it's exactly as you say, wild birds will behave naturally in the wild, and if you go and measure them in the lab, you are going to be measuring just a fraction of the behavior. That is exactly as you were saying. So if they're practicing the same song again and again, will they get the same time of dreams? Well, this is a Great question. So they practice exactly as you say, once and again during the day, the same song, and at night they have all these non-veridical replays. But we are just scratching the analysis of those. We find that among those practices, you have the ones that do sound like, that are correlated to the to the actual patterns used during the day, but then you have non-veridical things, and the non-veridical things we still didn't classify them to see what kind of diversity you have. So, but if you want, if you are still interested and you didn't lose all your patience, what I can share with you, maybe, is the following. It's a song that we put together for a bird, for an assigned bird, which is practicing over and over this song. So this is what this song is doing. This is what it's doing all day long, all day long. And what I'm showing you now is pasting together the sounds that corresponds to specific patterns that we could correlate with. So this is for a zebra finch, what it's dreaming at night, okay? Okay, completely different in structure. So it's very spectacular how it is so different, what they practice when they sing and what they replay at night.
Yes, sir. So I think there is one, I think it's a similar question.
Oh, there is there are studies where authors discuss that some animals have partial synchronized neural activity during sleep, chimera. Does bird brain also exhibit such states, either during sleep or singing? Well, this is a beautiful question for my coworker Anna, who is studying the synchronization of neural activity during behavior. So I'm going to try to summarize what I know of her work, but probably she would be better describing this. So what she finds is that when she measures activity with her student Tiama, when the bird is singing, there is a high level of synchronization between several neurons. How that synchronization is affected during dream, we don't know. So this is an open question. But yeah, there is a high degree of synchronization that allows them to recognize in the average activity of the brain regions very rhythmic and well-structured activity patterns. So there's a high degree of synchronization that allows in the average to survive these very rhythmic activities. So probably very soon they're going to be sharing their results, but for sure, seeing how this synchronization is affected during dreaming is a wonderful question. Yeah.
So if there are, I think there are no further questions. So I'd like to thank you, Professor Gabriel, for giving such a wonderful talk and for answering all the questions very patiently. Yes.
No, thank you very much for the invitation. It has been a treat and a pleasure to be here, and thank you all for your attention.
Yes. The pleasure
And
was
just a final announcement. If you want to travel to South America, Dynamics Days is going to be held in Buenos Aires from December the 9th to December the 13th. And it's a wonderful time of the year to go to Buenos Aires. So we hope you can consider it among your options this year.
Yes. I hope everybody has noted that. And yeah, so thank you very much once again. So there is one announcement regarding the seminar series. So the seminar series will go on a summer break for 3 months, and we will return back on September. So the details of the future talks will be shared with you shortly. So yeah, so we'll end the seminar here. Thank you, Professor Gabriel. Thank you everyone for attending.